The other tool that we developed is quantitative real-time PCR. So this is a PCR that's very sensitive, uses a TaqMan-based fluorescent probe. The nice thing about it is that once you set up the reaction and you carry out the reaction in the machine just reads it through the tube, the fluorescence, and you never have to open the tube. This becomes important when you are doing patient data, so you're not contaminating one patient's sample with another patient's sample. And I must tell you that not everyone does this. A lot of people in this XMRV world are using nested PCRs. Now, nested PCRs mean you have a set of primers that are called the outer primers. You do a PCR reaction, then you open the tube, you take out a tiny amount of that reaction, you put it in another tube, and now you use nested primers, meaning another set of primers inside, which gives it a lot more sensitivity and specificity. But in the act of opening the tube and transferring stuff, you can contaminate from one specimen to the other. And PCR being a very sensitive technique, you just need a tiny drop that you can't even see in order to contaminate this. So, my lab has stayed away from any nested PCR approaches, and we have stuck to these. The point of the slide was to tell you that this is an extremely specific PCR. So it only picks up XMRV. It doesn't pick up the MLVs that might be implicated, but it also prevents you from picking up mouse viruses, which are a source of contamination. I will come to it in a second. So, so you just have to know what tools you have when you use them. So here's our study. We looked at, this was done when we were still at Columbia University in New York. We looked at 233 consecutive cases of radical prostatectomy that came to the hospital at Columbia. And we looked at 101 controls. And to get controlled prostate tissue, you can't really ask a normal person because they're not going to say, yes, go ahead, take my prostate out. So these are all people who had benign prostatic hyperplasia, which is a condition that elderly men often develop. And they're treated with a transurethral resection. And so this was the transurethral resection sample. And we analyzed these samples by our quantitative PCR and by our immunohistochemistry using the antisera. And this is what the antisera shows you in the tumor. So remember I told you that the original papers showed that the virus was present in rare stromal cells, not in the malignant cells themselves. And here we see, we used a specific antisera that we generated. You can see the virus in the malignant cells. So here's an acinus of the prostate, and each of these is a cell, and inside each cell you can see this purple nucleus. So that kind of gives you a way to look for the cells. And each of these cells is just chock full of XMRV proteins. So everything that's brown is representing XMRV proteins. Here's this little inset that's been enlarged. You can see that same kind of granular pattern that I showed you before. So here is the virus present in malignant cells. It's a lot of variation, though, from patient to patient. This is one of our cases that expresses a lot of XMRV proteins. And here I'm showing you two other cases that are on the other spectrum that express very little XMRV protein. So this is one case. And you can see here, this is enlarged here. There's the same kind of cytoplasmic granular pattern, but not all cells have it. There are some cells that are completely clean, and then there are others that are expressing virus. And you can see the same thing here in this case as well, that again you have this granular pattern of staining, but then there are many cells that are cancerous but not expressing any XMRV proteins. So that's all consistent with, each one of these was counted as an XMRV positive prostate cancer. This is a summary of what we found. We found that 27% of prostate cancers tested positive for XMRV using either the PCR or the immunohistochemistry. So, we added everything. And then this is our controls. Our controls showed that 6% of people either had PCR positive or IHC positive for XMRV. And what does that mean? So either it means that our tests have a rate of false positive, that is 6%. That's quite acceptable for first generation testing for anything. The other possibility is that these are people with benign prostatic hyperplasia today, and maybe 10 or 20 years later, they are the same people who will get prostate cancer. And there's really no way for us to know this. These are all de-identified specimens. We cannot go back to look at them again. And the third possibility is that when you do a transurethral resection, you're taking out the tissue from around the urethra. That's not where prostate cancer develops. Prostate cancer usually develops in the periphery of the gland. So these might be people who already have prostate cancer, and we wouldn't know it at the time that these samples were taken. So there are many explanations for this 6%. But the important take-home is that 27% of the cancers tested positive for XMRV. So for the first time, we showed that there was a difference between the test population with prostate cancer and the controls. We also showed that the more aggressive the tumor, and Gleason grade is a way of assessing aggressiveness, the more aggressive the tumor, the more likely it was to have XMRV. The other thing we saw was that we didn't see any association between either the stage of the tumor or the age at diagnosis. And finally, what the previous paper had shown that there was a strong association between RNase L genotype and XMRV, we didn't see any association at all. And now there are many, many other studies that have come out showing that this association is not seen, and the original people also now say that that's probably true. So here is the cancer people and control people with the 3 different genotypes, and there's really no difference between cancer with XMRV, cancer without XMRV, controls with XMRV, controls without XMRV. They all have the same frequency of alleles. What is the implication of that? The implication is that the whole population, and not just the 10% that's homozygous for this allele, is susceptible to XMRV infection. So our original assumption that it'll be a small fraction of the 10% of people who have this allele is probably not true, that we are probably all, irrespective of our RNase L genotype, are susceptible to XMRV in the same way, or we don't yet know what makes us more susceptible. So to summarize, 27% of prostate cancers have XMRV. More aggressive tumors have a higher chance of containing XMRV. Virus is found in the malignant prostate cells, making it consistent for an oncogenesis model, and we see no association with a hypomorphic RNase L allele. Some general lessons that we've heard from this, and this will be important if you read the literature or you read reports of what somebody's summarizing about the paper, if you keep these things in mind. Tumors contain very tiny amounts of virus. I mean, these are really tiny amounts. And I think that that's going to be true of any pathogens that we find from now on, because I think a lot of the pathogens that were present in abundant amounts have been already discovered. And now is the era of finding either the pathogen that's present in very small amounts, or the pathogen that may be present in abundant amounts, but there's a particular genotype variant of it that causes disease. And here I'm thinking of the norovirus causing IBD. There's a particular kind of norovirus that causes IBD, and the others don't. So I think those are the kinds of things you should be thinking about. The other thing is that contamination from mouse tissues can occur very easily if the same microtome is used to cut human and mouse tissues. Now, many research labs process mouse tissues on the same machine that they process human tissues, and we found that that was a huge problem in our initial few things that we just had to discard about 100 samples that we cut originally because of mouse contamination. The other thing to remember is that deeper sections cut from the same tumor may have very different amounts of virus than the first few sections. And detection of the virus by immunohistochemistry has not worked well in tissue microarrays for us. and full sections are the best, which just makes everything much more expensive and time-consuming. But it also helps you understand that somebody doesn't find the virus, it may be because they were just focused on tissue microarrays, which are these small, very small pieces of tissue. If you go to my lab website, which is given here, there's an XMRV website which has a lot more information on publications. So this is a page that gets an automatic update no matter who publishes on XMRV. This page gets updated with the publications on it. And then you can find out more things about XMRV. So here are protocols from my lab that we've published and made available to everyone who wants to use it. And these give a little more detail than what you can put in a scientific paper, and so on. So not everybody agrees that there is XMRV in prostate cancer, and there's a bunch of papers that have come out saying that they don't find the virus in prostate cancers. And many of them use tests that they've developed. There are no good control samples that are known positives or known negatives. So it's really hard to say if these people would have picked up prostate XMRV from any cases or not. You know, it's just hard to assess. So this may be real. This may be real that in Europe there's no XMRV in prostate cancer patients or not, and the jury's still out. And so there's a lot of variation. And so here's a paper from Emory University that does find XMRV in prostate cancer, and their number they came up with is exactly 27%. And Jason Kimata finds XMRV in 25% of prostate cancers. And Ike finds it also in about 20% of prostate cancer. So those who find it are roughly finding this 25%, and those who do not find it, you know, are coming up with 0. So we'll see what happens with that. Last year in October, there was a paper that I was very surprised to see, that there was XMRV in chronic fatigue syndrome. Okay. And they identified XMRV DNA in 67% of patients and on only 3.7% of healthy controls. Okay. So, and again, this is controversial. So there are a few other studies which failed to detect XMRV. And there's, you know, mostly from Europe. And there was a more recent study from the NIH and FDA, which found XMRV-related viruses. So